Okay, I think we can go ahead and get the ball rolling here. So welcome everybody. Thank you for coming to today's orientation session for the CompuCell virtual tissue modeling workshop and hackathon. I am Hayden Fennell. I'm the coordinator for the workshop this year. Normally, Professor James Glazier, who also is hosting the workshop, does these orientations, but he is currently in South Korea at a conference, so I'll be delivering your orientation today. Just as a quick note, this session, as well as basically all of the workshop sessions, will be recorded and distributed on YouTube for people to view after the fact. Most of the workshop sessions will be live streamed as well. This session is not currently being live streamed, but it will be uploaded online after the fact for future viewing. Just so everybody is aware the session is being recorded. This slide deck is available in the student materials folder in the course Google Drive. I will paste that link into the chat here so people can go grab the slides if they would like to. There's a subdirectory called orientation that has this presentation in both PowerPoint and PDF formats. Right now, the student materials folder is kind of sparse. It's just orientation and a resources folder right now, but that folder will be populated as we prepare for the workshop over the next month with module slides, hackathon teaming information. So do keep an eye on that. Whenever there's large updates or things that you need to look at, I will send out an announcement by email as well. But this folder is where you will find basically all of the materials and resources that you'll need over the course of the workshop. You can submit any questions, concerns, or suggestions today through the Zoom chat. If you have questions along the way, please feel free to use the raise hand function in Zoom. And we can stop and ask. I'll have a couple of points throughout the presentation today too, where I'll stop for time for questions. So don't worry about that. We also have a Slack channel set up for this year's workshop, or Slack workspace. I'll go ahead and put this in the chat as well. We will have a shortened tiny URL for the Slack closer to the time of the workshop, but Slack's share URLs are only good for 30 days. So we're waiting until a little bit closer to the workshop to get our shortened tiny URL version. But for now, I've put the Slack link in the chat. We use the Slack for a lot of side communication throughout the workshop, so it's a great place to ask questions that maybe aren't directly relevant to what's happening in that day's content, but that you still need answered. We can get to that kind of on the side using the Slack channel. The Zoom chat is generally reserved for questions that are immediately relevant to the content being discussed. Slack is also good for hackathon teaming, but we'll talk a little bit more about that in a later slide. If you have not downloaded and installed CompuCell3D to your personal computer yet, that's okay. We're going to have a short demonstration at the end of the orientation today on how to build a basic simulation. But if you don't have it installed, that's fine. Again, these slides in the recording will be available after the fact if you wanted to try it on your own time once you do get it installed. If you're having trouble getting CompuCell installed on your computer, or if you're running into issues, or if you just haven't tried yet and would like some help walking through the process, we do have our second installation help session tomorrow, July 2nd, from 1 p.m. to 5 p.m. Eastern Daylight Time. Which is the same time zone as New York City. Make it a little bit easier to figure out the time zone stuff. If you can't attend that session tomorrow and still need help with the installation, please send me an email as soon as possible and we can set up an appointment to take a look. We will not be offering, I'm going to put my email in the chat here. We will not be offering a remote server option for running CompuCell this year. So it is important that we get everybody a copy of CompuCell installed on their local computer before the workshop starts, because there really won't be much time once the sessions start for troubleshooting installation issues. And we'll talk about what the daily schedule looks like a little bit later. So please do contact me as soon as possible if you can't get CompuCell installed on your machine. And we will make sure that you get a working installation before the workshop begins. Okay, so just a quick outline of what we're going to be doing today. We're going to just go over today's sort of an informal introduction to what the workshop's going to look like. We're going to talk about course logistics, a few areas where you may want to do a little bit of background reading in preparation for the workshop. It's not necessary that you do preparation ahead of time, but it is helpful sometimes if you've at least seen a couple of these things here and there before you start. So we'll talk a little bit about what to look for and where to look for it. We're going to talk about the hackathon logistics, what that's going to look like, offer some resources on where you can find more information, and then we'll do a quick run through of basically how to build a very basic simulation in CompuCell to kind of get you started if you wanted to start tinkering with it before the workshop starts. And we'll have some time for questions at the end. We do have more than 150 people registered for the workshop this year. That doesn't, typically we don't have everybody stick around for the duration of the workshop, but we do have a lot of people involved this year. We'll still do our best to support everybody, but please do try to be a little bit patient, especially with email requests once the workshop starts. We tend to get a lot of emails throughout the day over the course of the workshop, and we do try to have a policy of responding within 24 hours. So if you haven't heard back from us about something within, I would say 36 to 48 hours, feel free to send your email again, both leading up to the workshop and during the workshop. Don't be afraid to bug us a little bit, because sometimes in the huge mess of emails that we get for the next couple of months, things can get lost. So if we haven't responded within a day, please don't be afraid to send your email again. If you are an experienced user of CompuCell or have used CompuCell before and you would like to help with either assisting during sessions or potentially running a hackathon project, we are interested in bringing you on board if possible. So please do contact me as well if you're interested in helping with the workshop in a more official capacity than just attending it yourself if you've already got some experience with CompuCell. So again, welcome. I'm just going to go over a little bit of what CompuCell can do before we start talking about the actual outline of the course, just to give you a feel for what's been done in CompuCell in the past and if it's a good fit for what work you're trying to do. Basically, the bulk of what CompuCell is used for is for looking at behavior of cells within multicellular systems of all different types, and largely looking at the effects of different inputs on disease outcomes. So we look at a lot of different developmental biology applications, including developmental diseases. If we can identify the mechanisms behind how certain developmental diseases progress, we can potentially target therapies towards that. CompuCell is also used at looking at infectious diseases. So how do pathogens and hosts interact? How does disease spread through cells in a tissue? That also gets into some of the toxicology stuff. So certain compounds cause damage. What damage do they do? What determines susceptibility to damage? Do different combinations of compounds cause different patterns of damage? And ultimately, this is getting at kind of medicine and bioengineering questions. If we can predict behavioral outcomes using computational simulations, then maybe we can develop, more well-targeted drugs and treatment plans to kind of prevent issues that are typically only, diagnosable and fixable once the disease has got to a certain point. So basically adding some predictive power to our medical research. What CompuCell looks like in terms of scale. So CompuCell is a multi-scale agent-based modeling platform. If you think about sort of the grand scale of things, we start with kind of like the whole animal, which is larger in scope than what CompuCell does. And then you get down into individual organs within the animal, tissues that make up those organs, and then groups of cells, so the multicellular part, groups of cells within those tissues. And then you get even into finer grain detail of cell signaling between cells, individual cell behavior, and then even subcellular behavior of components within cells, just kind of decreasing scale in terms of the largeness of what we're looking at. CompuCell is sort of situated in this dotted line area here, somewhere between the organ and the multicellular scale. So typically we look at multicellular and tissue scale. CompuCell is capable of modeling whole organs depending on the size of simulation that you would like to run and the size of the organ. CompuCell is also capable of some intercellular signaling using some add-on packages that we'll talk about during the workshop. So you can do network modeling with CompuCell, but it requires the addition of other packages. CompuCell itself is mainly intended for modeling these multicellular tissue simulations. Give you some examples of what maybe some of that looks like. Some things that our group has done in the past with CompuCell. We have a model of, for example, polycystic kidney disease, looking at the development of cysts on kidney renal tubules due to, I believe this was a study in acetaminophen poisoning in the presence of alcohol. So you can look at the progression of cyst development on the tubule in 3D. Some other simulations we've got, tumor growth within a blood vessel network, looking at how the tumor interferes with the blood vessels, with its angiogenesis behavior, and how the tumor saps resources from the blood vessel network. A couple other applications. There's been a study using CompuCell on diabetic retinopathy therapy optimization. So this is looking at degeneration in the retina in diabetic patients, where you can take laser therapy basically and target certain portions of the tissue, damaged tissue for repair, and looking at different layouts of the laser treatment and the prognosis of healing over time with different laser treatments in the tissue. Kind of along the same lines, it's a study of retinal neovascularization, where the blood vessels in the eye actually start to grow into the retina. This causes retinal detachment. You can see here the green is the retinal tissue, and then the red is the blood vessel tissue vascularizing into the retina. And so if we can understand how the vascularization encroaches upon the retina, we can target treatment to kind of prevent this from happening. A few other examples of research outside of our group using CompuCell. CompuCell models of predicting disruption of blood vessel development because of various causes. So how the blood vessels are disturbed during development. You can see here, there's images of the blood vessels dissolving and kind of deaggregating, falling apart. Some computational modeling of genital tubercle development. So looking at malformations of the genitals during development and how to, again, target therapy to prevent the disordered development. And then there's also a pretty well-known model of secondary palate fusion and disruption. So how the palate fuses together during fetal development. And the study was kind of looking at causes for, or not causes for, but therapies for avoiding cleft palate disorder. So if the palate doesn't fuse properly, how does that happen, and how do we target therapies towards reducing the probability that that will occur? CompuCell's also been used in some viral infection modeling. It was particularly useful during the COVID period, where kind of switched over to modeling spread of COVID virus through tissue and through different organs. Not going to go into too much detail here about this one, but the idea is just to show you kind of a number of different things that CompuCell has been used to do in different published papers over the last 10 years or so. So a pretty broad spectrum of what you can do, kind of using the same general computational approach that CompuCell implements. So by the end of this course, our main learning objectives are that you will be able to kind of participate in the larger modeling process of starting by looking at a biological problem and identifying hypotheses and questions that you can ask and then answer with simulations. So coming up with addressable hypotheses that we can use CompuCell to study. Then we'll actually use CompuCell to analyze the components of multicellular systems and their interactions, build simulations out of those. And then use the simulations that we've built to generate data that we can display and analyze to kind of come up with biologically meaningful results from your simulation. Particularly if you're participating in the hackathon, you will get a good bit of hands-on experience of moving through this process. So course logistics. I guess maybe I should pause for a minute and ask if anybody has any questions so far. Feel free to put them in the chat or just unmute and ask. Okay. I think we'll go ahead and move on then, get through some of these details of course logistics, what the class is going to look like at the end of this month. So we will have links for 3 separate links, one for the Python bootcamp, one for the workshop generally, and then one for the hackathon. Sorry, I see a question in chat here from Ramon. I was wondering if there's any limit in terms of voxels for the CC3D simulation. Off the top of my head, I don't remember. I know that there is an upper limit in terms of like what you can do performance-wise before things start breaking. I think the maximum lattice size is something like 10,000 by 10,000 pixels. So 3D, that would be 10,000 by 10,000 by 10,000. But I can get back to you on that with specific numbers later, Ramon. You can make pretty large simulations, though. It's going to take a long time to run if your simulation is too huge. CompuCell can be run on, just a quick note, CompuCell can be run on supercomputing clusters. We won't be talking about that during the workshop, but if that's something that you're interested in, if you do have a simulation idea that's very large, you can break your simulation up into chunks and have it run in parallel on a supercomputing cluster. So we'd be happy to work with you one-on-one with that, Ramon, if that's something that you'll need down the line. Okay. So we'll have 3 separate links, one for the Python bootcamp, one for the workshop, and one for the hackathon, so that people can register separately for those 3 things. We don't have those links set up yet, but they will be set up within the next week or 2, and we'll send those out by email, and they'll be posted on the website page as well. So you'll have a permanent way to access those in addition to emails, because I know these emails tend to get lost sometimes. We'll have a link to a hackathon teaming document that we use to help people find hackathon teams. So if you are interested in participating in the hackathon, we'll have that up by the end of the week and a link sent out for that as well. And these things will also be posted to the Slack channel, which I will now, let me repost that because I know a few people have joined since we started, or let me add these to chat. So this first one is a link to our student materials folder where you can find all of the resources you'll need for the workshop. And then here's a link to the Slack channel. And there will be a dedicated hackathon channel in the Slack workspace that you can use for teaming. And we'll talk a little bit more about hackathon teaming and how that works during the workshop itself. I've got a few notes today, but we'll get more into that during the actual workshop later. So Zoom logistics, I think everybody is pretty well versed in using Zoom at this point. This is an old slide from when we were originally switching to Zoom during COVID. But just a few important notes, screen sharing and microphones will be disabled by default when you join the session. This is mainly just so that, you know, the large number of people, we don't get too many conflicting microphone signals. But you will be able to unmute. So if you do need to ask a question, just feel free to raise your hand and we can, if you can't unmute yourself, we can unmute you and you can ask, or you can always feel free to put it in the Zoom chat. For each session, there will be an instructor and essentially a moderator in the chat who will be actually monitoring the chat. The instructor themselves will have certain check-ins where they'll check the chat, but they won't be checking the chat in real time. So the moderator will be the one actually reviewing questions. Some questions can be answered directly in the chat, but if it's something that we feel especially pertinent to what the instructor is talking about, or if you would like the instructor themselves to address it, we can break in and ask them during the session. For some of the activities, we'll have breakout rooms where you can join to get some one-on-one help. We won't be doing breakout rooms during the delivery of content itself, but during some of the activities, we'll have breakout rooms where you can go to get some extra help if you need it. If you have any technical problems with Zoom, please let us know right away so we can help you get Zoom installed and updated. That is one thing to be aware of, is that you do want to keep your Zoom client updated. I would say probably try to update it the day before the workshop, and then maybe every couple of days during the workshop. Zoom updates tend to be a little bit unpredictable. And sometimes if your client is out of sync with the version of whoever is hosting the meeting, some features don't work, particularly things like polls, reaction emojis. There's a number of things that are deactivated if your client is out of date. So polls especially are important because we use polls to gauge progress throughout exercises. So you'll need to be able to participate in the polls. So make sure that you do have your Zoom client updated. And if we are made aware of a sudden update during the workshop, we'll make sure to send an email out about that and let everybody know to update their Zoom clients before the next workshop day. Videos and PowerPoints and exercises, including code and instructions, will be made available after the fact in the student materials folder, so you can continue working on them offline if you would like to, or so that you can maybe redo them later if you want to come back to this content after the workshop. Here are some links to our people. Again, Professor James Glazier will be the one sort of heading up the workshop with me. Some other important contacts to know: our main instructors will be Professor Gilberto Thomas, Pedro Sensi del Castel, and Julio Belmonte. And then Jim Sluka and TJ Sego will also be available periodically to help with some of the more technical stuff that we'll discuss during the workshop. A list of all the emails will be, again, included in the pre-workshop emails that we'll send out a couple of days in advance. So you'll have all of the necessary contact information. But if you're not sure who to contact about something, just email me. And if I'm not the correct person, I will forward you to the appropriate party. Quick overview of what each day of the workshop will actually look like. So they're sort of long days. We start at 10 a.m., again, Eastern Daylight Time, or New York time. And then we end at 6:30 p.m. So the first part of the day will be, we open the meeting at 9:30 a.m. We don't start till 10, but at 9:45, our instructors for the day and myself will be available for informal discussion, for if you had some issues the day before, troubleshooting, maybe some code, or if you have questions from the previous day's materials or questions about today's materials, you can ask it then. I know 15 minutes isn't a ton of time, but people usually don't have a ton of questions first thing in the morning. At 10 o'clock, we'll start our first module, which is 90 minutes. Each module is 90 minutes. There's 4 modules per day. At 11:30, 11:45, we'll have a 15-minute break, then module 2. And then we'll have a 90-minute lunch break, which is divided into 2 pieces. So there'll be an extra session, 45-minute extra session from 1:15 to 2, followed by 45 minutes of lunch break for everybody. The extra session is extra space that we use for, a lot of times we'll use it for hackathon teaming, if people want to give short presentations of maybe hackathon project ideas that they have. We'll fit that in there. We have some demonstrations sometimes. So these are kind of considered to be optional sessions. If you do need the full 90 minutes for lunch, you don't have to attend the extra session, but we'll give a schedule of what each extra session will be throughout the week, so you can attend if you're interested or if you need to. After lunch, we'll come back for our third 90-minute session from 2:45 to 4:15, followed by a 15-minute break, and then our last session of the day from 4:30 p.m. to 6 o'clock p.m. And then the last 30 minutes of the day, give or take, we're happy to stick around a little bit longer if we need to, if there's a lot of questions, but we do reserve the last 30 minutes of the day for, again, another informal discussion, troubleshooting, and Q&A session with the instructors from that day. So if you had any questions from any of the content over the course of the day, you can ask them there. So anything that wasn't answered during the session, or if you had a question come up later, you can ask it then. We know that 2 o'clock for those of us who are in the Eastern Daylight Time is a little bit late for a traditional lunch break. But we found that with all the different time zones that will be joining us, it kind of works out the best for various people. But if you do need to take a break for a meal earlier in the day, it's perfectly fine if you need to step away for a while. And we will have the live streams going. So if you do need to walk away, to get your meal, but you don't want to miss the session, you can always tune in on the YouTube live stream and watch it there if you can't bring your Zoom meeting with you. Any questions about the schedule, layout of the workshop? I guess I should mention, so we'll start the workshop on July 28th, which is a Sunday. That'll be our Python bootcamp day. Then we'll have 6 days of CompuCell workshop from Monday to Sunday. There will be a 5-day break after that before the hackathon. And then we'll start the hackathon on August 9th, which is Friday, and that goes till Sunday. That Friday will be kind of a half day for the hackathon, so we'll probably start sometime in the mid to late afternoon, mostly just to help people get teams organized, figure out who's working on what project, and then we'll start our major coding sessions of the hackathon on Saturday. Do have a question here. How long we have access to student materials after each day of the workshop and hackathon? Are there any deadlines for assignment submissions during the course? No, so I'll answer in reverse order. We will not have assignment submissions during the course. So there will be activities that you can participate in, but we won't be asking you to do anything outside of workshop time. The days are already long enough, and we know that people need to spend time with their families and eat dinner and things. So we won't be asking you to do any kind of outside work. You're welcome to work on the activities outside of the workshop time if you would like, but there won't be any requirements in that regard. And as far as how long you'll have access to student materials, those will be available perpetually moving forward. So things will be uploaded into the student materials folder each day. So you can go grab the new materials, but that folder will be accessible into the indefinite future. So you'll have persistent access to those as long as you have the link. The Slack channel, though, I think I get to this in a later slide, but the Slack channel will only last about 60 days after the end of the workshop. So anything that you need off of the Slack channel, you'll want to make sure that you grab it before the Slack workspace expires. But the student materials folder on Google Drive is permanent. All right, so I'm going to go over this a little bit quickly so that we have time for our demonstration at the end. But we have a few different background areas that we think are important to have some exposure to kind of before we get into the workshop. Those 5 areas for us are the general principles of modeling. Specifically for CompuCell, you want to know a little bit of Python programming, which is why we offer our boot camp. If you've never programmed in Python before, the boot camp is probably the best way to get prepared. We'll talk about that in a minute, though. Obviously, for computational biology, it's good to know a little bit of biology, but also some biophysics and a little bit of mathematics. Again, you don't need to know all these topics in depth, but you should know something about all of them. And if you don't, you will by the end of the workshop. But if you are interested in maybe spending a little bit of time reading up before the workshop, we've got some resources coming up for you. See, we have a question in the chat. I'll loop back around to that in just a second. So one of the things that we like to talk about with the background principles of modeling is just having some some idea of how to ask good questions that can be answered realistically with models. So defining questions that you can actually address with a simulation that aren't too large or the scope isn't too broad, selecting meaningful variables, so how to find starting parameters from literature, how to extrapolate parameters that maybe aren't immediately available in literature, how to decide which components and mechanisms are the most important to include in the abstraction of your model, a number of various things. So we'll have a couple of papers that I can recommend that might give you a good start on this if you've never done this kind of work before, to take a quick look at. As far as background in Python programming goes, so the Python piece is probably, in terms of preparation before the workshop, learning a little bit of Python is probably the piece that will cause the most friction if you don't know any yet during the workshop. So the basic things you need to know for Python are basic syntax and indentation. You need to know how loops work, for loops specifically, which will also entail some Boolean logic and conditionals. You need to know about object types, lists, dictionaries, what a class is, scoping and inheritance. Specifically for CompuCell, knowing what the self.dot class is useful. CompuCell uses the self.dot class a lot, particularly in reference to its cell list. And then you know a little bit about how to write attributes and methods within classes. Again, if you're attending the Python bootcamp, we will cover all of this for you. If you don't have time to attend the bootcamp before the workshop and you're unfamiliar with any of these concepts, we have a couple of places where you can go to learn it. If you're having trouble learning about Python on your own over the next couple of weeks, Python is one of those things that it's very easy to learn, but because there's so much content out there, it's also very easy to accidentally start learning all the wrong things. So if you do need some help learning a little bit of Python on your own and you can't attend the boot camp, please send me an email and we'll make sure that you have the resources that you need to get started. Our Python bootcamp recordings from last year and the year before are also all available on YouTube. So if you did want to go through it in advance that way, you can do that. This year's bootcamp will be a little bit different in format, but last year's bootcamp would also work. So a little bit on some things you might need to know for biology. So again, this slide, I'm not going to go through this in great detail today. But this slide is sort of a number of different prompts to get you thinking about things maybe that you have or haven't seen before. So what are cells? What do they do? How do cells generate forces? How do they move? How are tissues constructed? How do cells stick together? How do they kind of aggregate into different things? And then developmentally, how do they differentiate into different types of things? What are chemical reactions and regulatory interactions? Just a number of different questions you can ask yourself to maybe kind of get started on investigating a little bit of biology if you have never done any of the biology before. Again, you don't need to know any of this in great detail, and we will cover everything that you do need to know during the workshop, but we've found that it's easier to learn some of this stuff if you're at least familiar with some of the terminology. Sort of a surface level Wikipedia understanding of some of these concepts is perfectly fine. Again, background in biophysics, same thing. If you've never done any biophysics, you might just want to do some searching for some of the topics listed on the slide. I would say probably the most important thing for the workshop is understanding how diffusion works. Diffusion is one of the key components in how CompuCell functions. Again, we'll have a session talking about diffusion, but if you wanted to do some research ahead of time about the diffusion equation and how diffusion just works mechanistically, it would, help with making sense of the workshop content once we get there. Background in mathematics, the two pieces that you'll probably need the most background on for CompuCell is first order ordinary differential equations and rate laws. So again, we'll talk about these as necessary during the workshop, but having a basic understanding of at least what differential equations and what rate laws are going in will help a lot. Some places to find some of this background information. So this slide, I won't post all these links in the chat, but if you would like to access any of these, the slides are available in the Google Drive. We have some resources for each of the topics that we've discussed. On the Python front, there's this Rosalind Python Village 6-exercise mini course that we think is a pretty good assessment for the workshop. If you can do exercises 1 through 5, you're in pretty good shape for both the workshop and participating in the hackathon. And again, if not, we offer our boot camp to get you up to speed. And then we have some other resources here for each of these topics. If you have any suggestions for other good resources or reviews of these concepts that you find along the way, feel free to forward them to me. We may add them to our list of recommended things if we think they're a good fit. All right, so let me come back around to this question. In addition to the live streams, will the recordings of the sessions be available? Yes, so the live streams to YouTube will be processed once the live stream is ended, and then will be available on Professor Glazier's YouTube page going forward. Usually it takes a few hours for the live stream to process into a permanent video, but we'll send out links at the end of each day, or yeah, at the end of each day we do one email of end of day announcements and one email of morning announcements the next day, and the end of day announcements will always have the links to the live streams from that day in there. And then at the end of the workshop, we organize everything into a big playlist so you can actually just watch through in sequential order. So yes, the videos will be available after the fact. Any questions so far? Should stop for a minute and ask. Feel free to drop them in the chat as we go if anything comes to mind. I'm going to zip through this a little bit quickly so that we can do our CompuCell demonstration. This is just resources for the class itself, not so much learning resources, but where to find things relevant to the course. Again, if you need access to these, slides are available in Google Drive. Most things you need for the course can be accessed through the CompuCell.org website, including links to downloads as well as our help manuals. We have 3 sort of different manuals. There's a quick start guide for getting started with CompuCell. There's the CompuCell reference manual, which talks mostly about simulation setup and parameterization, and some kind of basics of how CompuCell works, cell properties, things like that. And then there's the Python scripting manual, which is a big list of helpful tips on how to do different things on the Python side, which we'll talk about in a minute, kind of how these 2 components of system specification and Python coding work together in CompuCell. All class files, like I said, lecture slides, exercises, demos, and videos can be found through the course Google Drive. And then we will also have a file share channel in the Slack for people to share files with each other. If we need to drop files in for people to access that way, we can do that too. And then announcements and links and updates to the schedule will be posted to the CompuCell 2024 workshop page periodically. And again, all announcements will also be sent out in email format, but we like to update the website so that you have permanent access to links and information. Again, Slack channel, like I said, it's a good way to ask questions that maybe aren't directly relevant to what's going on in the current course session. It's the best way to ask for technical help because we have a number of our instructors kind of monitoring the Slack on and off at different times. So it's not as immediate as the Zoom chat, but it is a good way to get help for things maybe outside the scope of what's currently going on in the workshop. Again, it's permanent-ish. It's only going to be available for about 60 days after the workshop. Changes to to Slack's terms of service have now made the uh, open workspaces. It used to be that you just couldn't add anything new after 90 days. Now they actively delete it after 90 days. So we'll have time leading up to the workshop, and then about 60 days after that for you to access whatever you need off of the Slack channel. And we'll send out reminders about 15 days before the Slack goes down. So you have a couple weeks still to grab anything you might still need. We find that a lot of interesting conversation happens on the Slack each year, particularly regarding the hackathon. So we do recommend that you join the Slack and try to check it, a couple times a day throughout the course of the workshop. Last note, quick reference guides are very useful. So these were sent out with your acceptance letter in the pre-class files folder. They're also available in the student materials folder here. We have a reference for Python, quick reference for CompuCell for basic commands and properties, and then a quick reference to Tellurium, which is the network modeling plugin that we'll be using in the second half of the week. And we recommend having these either printed out in front of you or saved somewhere convenient on your desktop so that you can access them quickly as needed when you're working on activities during the workshop. Have some extra readings, again, available in the student materials folder under the readings folder. We just have some different papers in there that have been done using CompuCell, some papers on modeling, things that you may want to take a brief look at before we get started if you're interested in learning more about kind of what's been done with CompuCell and how to think about starting your project if you're participating in the hackathon. So speaking of the hackathon, again, our hackathon will be taking place from August 9th to August 11th. So you'll have about 5 days after the workshop to kind of get started on your hackathon project. We'll do some teaming exercises during that week too to kind of get people matched up. And then you'll have 2 days basically to build a prototype model of your system. We'll have a couple of project pitch meetings. Some of those will be during the extra sessions in the middle of the day. And then we'll have a couple in that week before the actual hackathon starts. So these will be lightning talks of 3 to 5 minutes each for you to pitch your idea. Try to get people interested in your project. We provide a template for that, so you can structure your talk kind of around the main talking points that we think are important for teaming. We'll send that out again, in advance of the workshop, so you can get started on that soon. And then we will help we'll select a preliminary list of feasible projects. So we're not going to say no to a project, but what we kind of mean by this is that we will help you scope your project if it's currently out of the bounds of what CompuCell can do. We can help you scope your project into something feasible within CompuCell, and that we think can be prototyped within just a couple of days. We don't want to let you set off on something that's too big to accomplish over the course of the weekend. If you're not sure if you would like to participate in a team, but you're not sure what you want to do, you can choose your top 3 projects and we can help you find a team before the hackathon. Or again, if you'd like to just try to do that through Slack or through contacting people that you've talked to during the workshop, that's also fine. We will not assign teams, but if you would like help finding teammates or finding a team to work with, we can assist with that. The way the hackathon works is it's mostly just open coding time for the groups. We'll have a number of instructors available each day who will kind of circulate between the different groups to help answer different questions. We'll have check-ins in the morning and at noon with the entire group, so everybody will come back to the main meeting just to kind of let us know how progress is going, what help they might need. And then you can go back to your breakout rooms for your groups to continue working there. And then we'll have an evening report both days so that we can give you feedback going into Saturday morning and Sunday morning. We can give feedback. And then on Sunday evening, we'll have kind of a short round of presentations where people kind of show us the end result of their model. their model prototype. And then post-hackathon support with the CompuCell team is available. So if you're interested in continuing to work on your project, either as a publication, or if this is something that you were starting, hopefully to work on for a thesis, or even just a personal project, it doesn't have to be anything official. We're happy to help you continue working on that after the hackathon. If you don't know what to do for the hackathon, but you do want to do something to make use of the time with the instructors, reproducing an existing model is always a good way to get some experience. We also like to recommend that people replicate models in similar software. So there's another software called Morpheus that we're currently working on doing some kind of cross collaboration with, and having people work away at trying to rebuild some of those simulations in CompuCell is always a good way to gain experience as well. So we can help you find a project for the hackathon if need be. You can start posting hackathon ideas to the Slack channel now. There's a sub-channel called Hackathon. If you're already interested in sharing your ideas and potentially finding people to work with, then you can start doing that there. We'll have a structured idea sharing Google Doc set up by the end of this week. We'll have that link sent out and some instructions on how to use that document so people can start teaming in advance of the workshop if they'd like. And then we have a list of possible reproduction project ideas at the link listed here. It's just a big list of papers that we think are good candidates for reproduction in CompuCell. So you can take a look through some of those before the workshop if you don't have an idea already. Expectations of participants. We don't have a ton of expectations going into the hackathon, but we do, if you would like to lead a project, we do sort of expect you to have an idea of what you want to model and why. So try to come up with a hypothesis or two that you actually want to test in your system, so that we can help you target your project for the 2-day weekend. If you are interested in just participating in a project, and this is good for people who are also wanting to lead a group, we like people to have a shareable description of yourself and your skill set / background. So it doesn't have to be anything super long, but any skills that you think are relevant to model building, programming, working in a team. If you've got a strong background in biology, that's always good to know. So just a short list of things that you think are relevant to the current application that we can use kind of to help you find a group. Again, it's perfectly fine if you're a beginner. We don't expect any kind of background going into the hackathon. People have all kinds of skills that are useful to projects like this that maybe aren't immediately obvious, but then come out in the group work setting. Expectations you can have of us is that for those 2 days, we will be available, not necessarily 24/7, but for most of each day for questions and assistance throughout the hackathon. We want everybody to leave with a working prototype of the simulation that they're interested in. So any questions about the hackathon before we move on to a quick demonstration of how CompuCell works? I'll give people just a minute if they want to type in the chat. Okay, so again, if something comes up, if you think of something, feel free to put it in the chat. We can get to it at the end. Actually have one question here. Is it possible to model cell behavior inside a hydrogel using CompuCell3D? That's a good question. I'm not sure if it's been done. I know CompuCell's been used to model foams. That was kind of its origin point in the 90s. We actually have, so Gilberto Thomas is a He's done a lot of work on, he's one of our instructors. He's done a lot of work on foams. He would probably be the person to ask about that. I can follow up with him and see, but that would be an interesting simulation combining um, hydrogel structure with cells. So we can talk more about that moving forward, Shruti. All right. So if there aren't any other questions, we will move on to our demonstration here. So I'm actually going to, because we're a little bit short on time, I'm actually going to go through like 2 more slides, and then I'm just going to switch over to a view where you can watch me do the demo in CompuCell. And then the slides have instructions on how to reproduce this yourself if you'd like to do this after the fact. So if you don't have CompuCell installed already, that's fine. You can try this on your own afterwards. If you do have CompuCell installed, you can go ahead and open it up and get it ready and follow along if you'd like. But CompuCell is made up of 2 primary tools. So there's the player, which is what actually runs and displays your simulations. You can interact with a simulation that's running by changing parameter values in real time with player. And then we have another piece called Twedit, which is basically just a code editor. And this is where you'll be doing most of your work in CompuCell of developing the simulations. And what this demonstration does is kind of shows how those 2 pieces interact. Quick notes on installing running CompuCell. Again, if you're having trouble, please contact us right away. We'll have our installation help session tomorrow from 1 to 5. So feel free to drop in then, and it'll be at this link that you used today. I'll be sending out another email with the link later today, but it's also on the website. So again, the latest version is CompuCell version 4.5.0. I believe it's at revision 2. It might be at revision 3. So if you get revision 3, don't worry, that's the correct one. Executables are available at CompuCell3D.org/sourcebin with instructions on how to install on each of the 3 platforms. If you're running a like a custom build of Linux and you would like to compile CompuCell on your own, we do offer the uncompiled version. We don't recommend it unless you're a Linux power user already. It's usually difficult for us to help debug that because of people's custom Linux builds, but that is available if you would like to try to do it that way. Okay, so now what we'll do is switch over to full screen share. And I will go ahead again. These slides are available here with step-by-step instructions on how to walk through. Some of these images you'll notice have a slightly different interface. This is the old interface that we used to run through NanoHub. I did not replace the pictures because otherwise the interface is the same. So your interface may look slightly different in terms of style, but in terms of function, it will be exactly the same. So I'm going to go ahead and pull up my CompuCell here. So when you start CompuCell, you'll have 2 windows. You'll have a terminal and you'll have player. For those who haven't run CompuCell yet, this terminal has to stay open the whole time you're using CompuCell, so don't accidentally close it because that will close everything you're doing in CompuCell. Learned that the hard way a few times, absent-mindedly closing terminals and exiting out of CompuCell by mistake. The terminal is also the place to go if you've sent a command to CompuCell and you're not sure if it's doing anything. The terminal will have output kind of explaining what it's currently doing, help you figure out if it's frozen or if it's just thinking. So when we start CompuCell, we've got player here. Can everybody see my player window? Just want to make sure the screen share switched properly. Excuse me, I'm sorry, I got some scratchy throat today. Okay, so nobody's saying no, they can't see it, so I'll assume that we can see. So we'll start with player. From player, you can open a simulation file if you would just like to run an existing simulation and see it, see the output animation. We're not going to do that just yet, but I will show you how. If you go to open simulation, we have a folder called demos. This comes pre-installed with your CompuCell installation. So you can open this, and there's a number of different simulations that you can check out with CompuCell. You can look at the code. This is also a good way of testing to make sure that your CompuCell installation is installed correctly. If you can open one of these simulations and run it, then your CompuCell installed correctly. So we'll actually let's open one of these. We'll go to book checker demos, angiogenesis, and then you'll just open the .cc3d file in player. Sometimes it takes a minute to get started, especially the first time you open a simulation. After opening player, it's got to load everything into RAM. So typically, after you've opened your first simulation, it'll open the next simulations quicker. So you'll see up here it changed the file path, which means it's opened it. And if we hit play, the window sometimes resizes when you restart a simulation. You can see we have our angiogenesis simulation here of blood vessel growth from a clump of cells from the start. So I know we're about to go over time just a little bit, but I do want to show you how to set up a simulation very quickly on your own. This will take about 10 more minutes for people who have tight schedules, just so you have an estimate of how much longer this will take. So if we want to build our own simulation, not just look at one in player, we need to open Twedit. And Twedit is here. It's this notepad with a pen next to your Zoom features. So you just click that button. Again, it might seem at first like nothing is happening, so you want to check your terminal. And we'll just wait a minute. I've already run this a couple times today, so my commands might look a little bit different from yours. But again, sometimes it just takes a minute to boot Twedit back up. Try again. Okay. All right, let me just close CompuCell and reopen it. Actually, so you can also open Twedit directly. This would be a good way to do that here. From your CompuCell folder where it was installed, you can just open directly into Twedit. Again, it'll still open the same terminal that needs to stay open. Some of you, this came up in our last installation help session. Some of you may get a message when opening Twedit, did not find VS in registry. The compiler may not work. Don't worry about that. What that's telling you is that you don't have Microsoft Visual Studio installed. You do not need Microsoft Visual Studio to use Twedit. Okay. So this is our Twedit interface. So this is, again, just a code editor. Like Notepad++, if anybody's ever used that before, with some CompuCell specific features. So we have a couple different things up here. We have some CompuCell3D ML, which has some pre-written code that you can insert into your existing code. Same thing with CompuCell Python, some code snippets. We'll go over how to use these during the workshop. What we want today, though, is we want to start our own project. So up at CC3D project, we'll go to new CompuCell project, and this will launch our simulation setup wizard. which is the easiest way to build a simulation in CompuCell. The wizard will generate all of the necessary code files that you need to get started, and then you can just modify those files instead of having to write everything from scratch. So the first thing you want to do is set your directory somewhere where you know you have full write or administrator privileges. So somewhere in your user folder, probably one of the safest bets on your desktop, things like that. You don't make sure that your user account has full access to that folder. If you try to save it somewhere where you don't have full privileges, CompuCell will crash because it can't make the temp files it needs to run. So I've already got mine set to a safe path. And then you want to give your simulation a name. There's a couple things you need to know about naming conventions. Again, this is in the slides. Your simulation can't start with a number, so it needs to be an alphabetic character. You can't have spaces in CompuCell simulation names. You can use underscores, but no spaces. And then you want to avoid special characters, so no exclamation points. You want to avoid periods and dashes. You want to give it just kind of a simple simulation name. So I'm going to call this orientation growth sim, and then I'm going to add 2 because this is the second time I've done this today. You can end or mix in numbers, you just can't start with a number. And actually I'll throw in an underscore there. You want it to be Python plus XML, so we'll click next. First window in the wizard is the general simulation properties. So this is basically determining the structure and behavior of the lattice on which your cells are situated. We're going to leave everything as default for now. So we'll have a 256 by 256 lattice, 2D lattice, because it's just 1 pixel in the Z direction. We're going to leave our boundary conditions, lattice type, and everything else as it is. Again, we'll go over what all these features do during the workshop itself. Next window is cell type specifications. So this is where we set our cell types. You'll notice we start with medium. Medium is the base cell type. It's what everything sits in. So you need to have medium there. And then we're going to create 2 cell types. So we'll say cell 1. We'll click add. And we'll do cell 2. And then you can also just press enter to add your cell types. Once it's been successfully added, it'll be added to this table. We don't want anything frozen, so we'll leave all these unchecked. Chemical fields is next. We're not going to specify a chemical field for this example, so we'll just click next and it'll leave it out for us. And then here is kind of the big piece of the wizard. This is determining which cell property and behavior plugins you would like to include in your simulation. Again, we'll go over all these plugins during the workshop. But this is kind of how which chunks of simulation behavior you want to start out with. You can add these later using the drop-down menus like I was talking about, but this is what will be inserted into the code from the start. So for our current simulation, we're just going to do a basic cell growth simulation. We want contact because we want the cells to adhere to each other. We want growth because we want the cells to grow over time. You'll notice when you check growth, that some of the features on the constraints and forces disappear. It's because only some of them are compatible with some of the plugins on this side. So we click growth, we narrow down which volume and surface plugins we can use, for example. We want mitosis because we want the cells to divide when they reach a certain volume. And then we'll click volume local flex because we need a volume constraint to keep the cells at a certain max volume. It's going to seem like a lot. Now, after the first day of the workshop, this will all make perfect sense. So we're going to go ahead and click next. And you'll get this configuration complete. And congratulations, we've just built our first CompuCell simulation. It's that easy. So you'll notice it's added a project here in our project manager. We drop this down, see that there's 3 files. We double click on the project. It will put the 3 files up into edit. Sometimes it takes a second to load in. I apologize for all the throat clearing. And throat's gone scratchy after these presentations. All right, and you'll see that there are 3 files that it's generated. We have an XML file and 2 Python files. We'll just go from left to right here. We've got our XML file. What this is essentially, this is just a markup file that sets up all of our simulation, what we call parameterization. So initial setup of the simulation. So we have basic properties of the lattice. So we've got 256 by 256. Again, this is what you saw in the wizard. Let's just put it into XML code for us. Cell types that we specified. We've got cell 1, cell 2, plugin name, volume, center of mass, neighbor tracker. We've got our contact plugin, which specifies contact energies between all the cells. They just default to 10. We'll go through an example of how you can actually get cells to sort within a tissue by just using differentiated contact tissue values during the workshop, or contact energy values during the workshop. And then we have our uniform initializer, which generates the cells when the simulation starts. So this is all initial simulation values. Our first Python file is just the same as the name of the simulation. This is basically just the Python script that starts the simulation running. You will almost never have to make changes to this file. It's just necessary to kick the simulation off when you hit play. And then the other big file is our steppables file, and this is where all of the Python code happens. So once the simulation is running, the steppables file is what governs changes in the simulation over time. We won't get into what all of this means today because it's a little bit detailed, but that's kind of how the two pieces interact. So the XML sets the simulation up from the start, and then the steppables Python file makes changes to the simulations as it runs in between Monte Carlo steps. Again, we'll talk about this in more detail during the workshop. So if we want to actually see this thing run, this thing is currently runnable in its generated state. We'll go over to the CC3D file in our project manager, right click, and then just click open in player. So again, it might take a minute. The first time you load a simulation after opening a new instance of CompuCell, sometimes it takes a second for player to start. But it should start here in just a minute. Yeah, you'll get a little dialog window, which is really hard to see actually on this white background. So it'll open in player, and then the simulation will automatically start running for us. You can resize the visualization window. We've got two cell types, green and green, which is not the best color scheme by default. You can change it. We'll show you how to change that during the workshop, but hopefully these colors are differentiated enough that you can tell between the two different cells. And you'll notice that the cells started in a big blob, grew outward. And then if you focus on a specific region, you'll see that some of the cells start to get larger, and when they reach a certain size, they split into two cells, which is why you have this kind of undulating mixture of smaller and larger cells. And then you can steer some features of the simulation using the model editor in real time. So for example, if we wanted to change the contact energies between cell types, we could do that here just by double clicking and changing the value. Not going to do that right now because we're running short on time, but there's a number of different things you can change. And if you want volume to be changeable over the course of the simulation, you can add that to the steppables file. So that's a basic overview of kind of how CompuCell works and how to build basic simulations. So if you would like to tinker around with the simulation creation wizard a little bit before the workshop, that'll kind of give you a good starting point of how to think about this once we get started. Again, we'll show you how to do all of this again during the workshop. This is just a quick primer. So I'm going to stop up here. Just a couple of final notes. So again, there's more detailed instructions on how to replicate the simulation I just built in the slides. If you have any questions about that along the way, if you'd like to try this on your own, please feel free to send me an email. Happy to help answer questions. So we have details about how to use the controls in the slides, and then we even have some more detailed information on how different segments of the code work that you can take a look at in the slides here. So some last notes on what to do to prepare for the course. So again, just to reiterate, make sure you can launch CompuCell on your local computer. If you can't launch it, please come to the hub session tomorrow or email us ASAP so we can help make sure you get it installed correctly. You want to make sure you can load, edit, and save a CompuCell simulation and tweet it. That's the easiest way to check that everything works is, can you load one of the demos? Can you modify the code, and can you save it? If you can do all of that, then your CompuCell simulation or your CompuCell installation is working fine. And then again, if you would like to do any background reading, you can review some of the core concepts that we've kind of touched on a little bit today. We have a quick survey that we'll be sending out within the next week or two to fill out about hackathon interests if you're interested in participating in the hackathon. And if so, if you have project ideas, so keep an eye out for that in your emails. And if you are interested in thinking or participating in the hackathon, please do start thinking about your project ideas or what kind of project team you might want to join before the workshop. All right, so that's all I have for today for the orientation. I'll stick around for a few more minutes for questions if anybody has any questions or concerns about anything we've talked about, or any questions about the format or what they need to do before the workshop. Yep, you're welcome, Liam and Rita. Yeah, if you don't have any questions, you are free to go. Just keep an eye out on your email for the next couple of weeks. And I would recommend periodically checking in on the CompuCell website for updates. We will be putting, last thing to mention, we will be putting the final schedule for the workshop content on the website within the next couple of weeks. We're currently working on getting all of that finalized and nailed down this week and next week. So at least 1 week before the workshop, we will have a final schedule of content posted to the website so that you can kind of get a feel for, if you can't attend every single day, which days you might want to prioritize.